Have you ever wondered if we could really bring dinosaurs back to life? Well, this concept, known as de-extinction, has intrigued scientists and the general public alike for decades. It's the idea that we could potentially revive these long-extinct creatures, much like the plot of the famous Jurassic Park. In the movie, scientists extract dinosaur DNA from mosquitoes preserved in amber, a concept that has since sparked real-world attempts to do the same. But unlike what Hollywood might have us believe, the actual process of extracting ancient DNA is fraught with challenges. For starters, DNA degrades over time, and the chances of finding complete dinosaur DNA strands are extremely slim. Even if we were to find a well-preserved specimen, many other factors, such as contamination and decay, make the extraction process incredibly difficult. So, extracting dinosaur DNA from amber-preserved insects may not be as simple as the movies make it seem. Scene script. Let's say we do have a sample of dinosaur DNA. Now what? In the world of Jurassic Park, they make it seem simple. Sequence the DNA, clone it, and voila, a dinosaur is born. But let's dive a bit deeper. DNA sequencing is the process of determining the exact order of the 4 chemical building blocks, adenine, guanine, cytosine, and thymine, that make up the DNA molecule. It's like reading a book, but instead of words, we have these 4 letters. Now, consider cloning. It's essentially creating an exact genetic duplicate of an organism. Sounds easy, right? But in reality, it's an intricate process that requires a viable egg, a surrogate mother, and a whole lot of luck. And here's the catch: dinosaur DNA is millions of years old. It's fragmented and incomplete. Today, we can't even sequence the genome of a mammoth, let alone a dinosaur. Even with a perfect DNA sample, recreating a dinosaur is not just a matter of copy and paste. Now, what if we could fill in the gaps in the dinosaur DNA with frog DNA, just like in the movie? Well, the idea is tantalizing, but it's not as simple as it sounds. The concept of using DNA from existing species to fill in the gaps in the dinosaur genome is called genetic splicing. However, it's not as straightforward as piecing together a puzzle. DNA is more like a complex recipe, and changing just one ingredient can drastically alter the final product. If we were to splice frog DNA into a dinosaur genome, the resulting creature would be just as much frog as it is dinosaur. In essence, we'd be creating a new species entirely. Moreover, DNA isn't just about physical traits. It governs behavior, metabolism, and a host of other biological functions. Therefore, mixing DNA from different species could lead to unpredictable and possibly dangerous results. So, creating a frog-dinosaur hybrid might not result in the prehistoric creatures we imagine. So, is there any hope for seeing a live dinosaur again? Well, let's delve into the reality of de-extinction. Currently, scientists are making strides in this field, with one of the most notable efforts being the attempt to revive the woolly mammoth. However, dinosaurs present a unique challenge due to the vast time that has passed since their extinction, making it nearly impossible to find viable DNA samples. Despite this, the journey to understand dinosaurs is far from a wasted effort. While we may never see a T-Rex strolling in our parks, the research conducted in trying to decipher their genetic code is invaluable. It provides us with rare insights into extinct life and the course of evolution over millions of years. We are learning more about the intricacies of life itself. In the end, while Jurassic Park may remain in the realm of fiction, the science it's inspired continues to push the boundaries of what we thought possible.